Okay, I think we can start. Hello, everyone. Thanks for joining the second tutorial on MEGAN6. My name is Simone Betz. As our first tutorial last year was such a great success, we decided to continue with it and bring to you another tutorial, this time covering different topics. Before we start, some administrative information from my side. This tutorial is being recorded and the replay will be available afterwards on the Computomics YouTube channel. If you have any questions throughout the tutorial, please post them into the Q&A window. We will have a dedicated Q&A session at the end. I'm very happy to announce Dr. Oliver Deutsch, our presenter of today's tutorial. Oliver is Scientific Product Manager for Metagenomics at Computomics. He has worked in industry and academia in multiple countries and has more than 15 years experience in bioinformatics, including more than 10 years with metagenomics analysis. In this tutorial, Oliver will give a quick overview of the recent updates to MEGAN6 Ultimate Edition. Then he'll dive into a live demonstration showing data visualization and how to automate workflows with MEGAN scripts. As mentioned at the end, we plan to have a Q&A session. Now I'd like to hand over to Oliver. Oliver, the stage is yours.
Thanks, Simone. So yeah, welcome again from my side. Happy to host this second tutorial. So as Simone said, the first one was quite a good success. I think we've had more than 800 views on YouTube now. So it's proven quite popular. So there's definitely demand, and this is why we're doing a second one now. Yeah, without further ado, let's start with the tutorial. So what I'd like to cover today, and this is also partly based on the input from the questionnaire that we sent out. So we've taken some of your feedback into account. I'll do a quick presentation on Computomics and MEGAN6. So where is Computomics involved? Then I'll show you some updates to MEGAN6, what's new, what's different. But then most importantly, I'll do a live demonstration of data visualization features in MEGAN. And then I'll also do a live demonstration on how to automate workflows with MEGAN scripts. And then at the end, we'll have a Q&A session, and I'm looking forward to your questions and feedback. So let's start the presentation. A very quick overview of Computomics, the company that Simone and I work for. So Computomics was founded in 2012, so we're almost 10 years old now, founded by experts in machine learning, plant research, and bioinformatics from the Max Planck Society, the ETH Zurich, and also the University of Tübingen. In those 10 years, we've completed more than 180 projects for customers in 15 countries. So even though we're based in Southwest Germany, our customer base is very international. We apply machine learning to genetics, different omics data sets, phenotype, microbiome, and environmental data sets. And our customers are from biotech, food and beverages, consumer products, and also plant breeding. Now, in the metagenomics space, we have those three pillars here. So we have MEGAN, the interactive metagenomics analysis tool. We have Morpheus, the platform for metagenomics analysis. And then we also have custom projects. MEGAN6, you'll be probably familiar with, is a standalone GUI-based software for metagenomics analysis. The Ultimate Edition contains KEGG pathways and also scripting functionality, which is very useful. But MEGAN always requires some data pre-processing. So you need some bioinformatics skills and also some compute resources to align your FASTQ reads against the reference database. If you don't have bioinformatics knowledge or you don't have the compute resources, you can use Morpheus, our platform for metagenomics analysis, where we integrate all the pre-processing, so you don't need to worry about the bioinformatics. And then the output is fully MEGAN compatible, but also the platform itself contains some data visualization and data storage features. On the very right-hand side, there's custom projects where we work with clients to understand their research question and then analyze their data for them, deliver a written report, and ideally this will then contain actionable results that our clients can use. But today we're going to focus on MEGAN6. So MEGAN6, to my knowledge, is the world's only interactive metagenomics analysis tool. And depending on obviously your input data and also the reference databases available, you can go down to strain level resolution with MEGAN. It's been cited more than 4,000 times since its release, and more recently, long read annotation has also been integrated. So if you don't like short reads and you're working with PacBio or Oxford Nanopore data, this is compatible with MEGAN as well. MEGAN contains command line tools, for example, the Meganizer tool, but the most common use case is really to analyze and visualize metagenomics data in an interactive manner on a normal desktop or laptop computer. MEGAN also has some data export and import functionality, which can be really useful, and we'll cover that in this tutorial a bit. And then the Ultimate Edition also has the KEGG pathways and command line and scripting functionalities. And we'll touch on that as well. Here highlights the main differences between the free community edition and the MEGAN, so the Ultimate Edition. The biggest difference is that the Ultimate Edition contains KEGG pathways that are up to date. And for that purpose, we license the KEGG database from Pathway Solutions Limited to make it available in MEGAN. And what this means is that you can really go from taxonomic analyses to functional analyses of your shotgun data without having to perform any additional intermediate steps, basically. And you can use the most widely used pathway database straight away. There's other differences. So, for example, the Ultimate Edition has extra plots, and also the Ultimate Edition has additional command line tools and the scripting functionality that we're going to cover in today's tutorial. Okay, so just a couple of slides now. This is a typical screenshot of MEGAN looking at the taxonomy view. I just don't want to go into detail as we'll cover this in the interactive part. Also, KEGG view that you can use to compare functional pathways. So here in this case, this enzyme here, for example, is only present in the blue and in the yellowish sample, but is not present in the middle two samples, in the green and in the brown one. So you can use this to start generating hypotheses, or maybe testing hypotheses that you already have around the functionality of your microbiomes. Okay, so before we go into the interactive part of this session today, just the most recent updates of MEGAN. So last week or 2 weeks ago, MEGAN 6.22 was released, quite a major update. Now we have a current up-to-date KEGG release, so February 2022, that also comes with new mappings of those KEGG enzymes to NCBI accessions. And basically this comes with every new MEGAN release, but what's really new this time is that we've greatly improved the mapping of those NCBI and R accessions to KEGG. So in this example here, going from 90 million to 19 million to 60 million. And the way this was achieved was using NO3 instead of a direct mapping. And what this really means is that with functional annotations, what we usually struggle with is that the annotation rate is often a lot lower for functional annotation than for taxonomic annotation. And hopefully with this improvement, we're bridging that gap a bit and improving or increasing the annotation rate on the functional level. Also with the current release, we have full M1 compatibility. So if you have a newer Mac with Apple's own M1 processor, MEGAN will now work properly. There's a new splash screen and icons. So you've probably seen the MEGAN icons in the intro of this presentation. So they're also now in the software integrated. And there's a couple of under the hood improvements that you probably as a user won't notice. So now we've completed the slides, and now I'd like to start with the interactive part. I'll stop this presentation and I will share my screen. And so you should be seeing my desktop now. Not much on it except from a terminal. And what we'll do now is we will start MEGAN. Okie doke. So, okay, so the first bit on today's agenda was interactive data visualization in MEGAN. So if we want to do this, we need some data, and I have a data set that I like to use for demonstration purposes because it shows has a couple of differences between groups, which is always nice to demonstrate. So I will open this data set, which is 36 samples of fecal samples from kittens that were fed to different diets. And so we've loaded the sequence data, or rather the MEGAN compatible comparison file here. Now those comparison files are a lightweight way of working with your shotgun data. So instead of having 36 samples, like gigabytes of alignments, this is a megabyte sized file that contains everything except from the reads and the alignments, so has all the counts. So for most demonstration purposes, this is perfectly sufficient, and it's really easy to work with this kind of data. Okay. But what we really need before we can do any meaningful visualizations is we need some metadata. So we'll import some metadata as well. And the metadata is essentially a description of, and you'll see this in this window here, is essentially a description of each sample and some metadata columns. So in this case here, it's diet. So there's two diets, this one HPLC and HPMC. And this is what we're going to use to color our samples with. Now, if you want to have a quick look at how the metadata looks, it is simply, I'll just open the file in the text editor. It is a simple text file with those headers here and tab delimited information in it. Okay, so we've loaded our metadata now. I'm going to choose a color scheme that is a bit more screen friendly for this tutorial. And what we'll do now is we will use the diet. Oops, we'll use the diet to color our samples. So use to color samples. And now we've got two colors here that are easily distinguishable, so gray and orange. And now what we can also do is we can sort by this column. And this makes it even easier. So here now we have one diet group on the left, the other one on the right. And this makes comparisons much easier. Okay, so we'll just start off with a couple of easy things that we can do in this taxonomy window. We've covered this more in depth in the last tutorial, so I don't want to go into too much detail. But what you typically do here is you go to your tree and you collapse your tree at a, oops, no, wrong. And you pick a taxonomic level of interest. Let's say this is the genus level. And then on the left side, on the right side, so the leaves of this tree are now the different genera, and the bars here are the differential, or the abundances of those genera across your samples. And what I like to do with this study, because I'm quite familiar with it, I go and have a look at Bifidobacterium, because I know it only ever occurs in this group on the right, in this side group, and not in the other one. So just to make sure that during the import process, everything has worked. So it's worked, I'm happy with this. And what we can do now is we can collapse or uncollapse this tree and compare the abundances of our taxa at different phylogenetic taxonomic ranks. Now, and each time you collapse or uncollapse, MEGAN will automatically update the counts for the specific level that we're looking at. And this is a really nice way of comparing your samples. And you can, like, even from this top level view here, you can immediately spot taxa that are highly present in one of those groups and not present in the other. Okay, but we wanted to do something different today. But before we look into the other plots or the other graphs that we can do, I just wanted to have a quick look at the KEGG functionality, because this is also very useful. And KEGG as a database is quite nice because it also has this tree-like structure where you can uncollapse and collapse. So in this case here, and we always have very handy. So on the very right-hand side, we now have individual enzymes, and those enzymes always start with this K number, and they nest in biochemical pathways. So this one here with this KO number is glycolysis, and then those pathways again are nested in those pathway groups like carbohydrate metabolisms and then metabolism. So this is quite handy, again, if you want to calculate abundances on different levels. But what most people like to use KEGG for is if you have a pathway of specific interest to you, you want to look at the pathway map. So you click on Show KEGG Pathway Map, and then this pathway map will appear. And now those numbers here, those are enzyme commissions, and the bars in those boxes represent the abundance in your different groups again. So you can do two things with this visualization. So one is you can look for pathways that are complete. So where you have enzymes across the whole pathway. So you can hypothesize that this pathway is functional. But you can also use this to look for enzymes that are differentially abundant. And for example, I can't see one in this here that is only ever present in one, but for example, this here, the orange bit is a lot larger than the gray bit. In this one here, there's only orange and hardly any gray. And you can use the different scaling here. So the counts are currently scaled on a square root scale. You can use a linear scale or maybe a log scale to make the differences more striking. So here it actually looks like there's hardly anything for this enzyme 5424. There's hardly any presence or almost nothing for this enzyme in the gray group. So this can be a really nice starting point to generate hypotheses on about the functional potential of your microbial communities. And it can also be used to test hypotheses that you already have on what you think that your communities are doing and why they are different in different habitats or different conditions. Okay, so we've okay, let's put the KEGG pathways to the side for now and go a bit more in depth with the other visualizations that we wanted to look at. So, because we wanted to cover the charts, the different charts that are available in MEGAN. But before I do that, I want to start with the cluster analysis, because the cluster analysis, I think, is one of the most important steps that everybody does. And like almost any paper on microbiome data analysis does contain some sort of cluster analysis. Okay, so before we do this, there's one key thing to remember. So all the plots or charts that I generate now, they depend on what's currently selected in this main view. So at the moment, there's nothing selected here. So if I now just do a chart, let's just do the cluster analysis, it will automatically select everything, because otherwise it wouldn't be able to produce this chart. So this is really important to keep in mind, because currently we're at the order level of the taxonomic hierarchy, but for a PCA, we probably want to go to the species level or maybe the genus level. So we select this appropriate level, and then what we do to make sure, we select all leaves. So basically all those genera. This is now very difficult to visualize here because it's so many species. And yeah, so now if we do the this cluster analysis, it will use the differential abundance, or no, it will use the abundance information of those species to generate this PCOA plot. So the first thing that we can see here is that the samples from this one diet group colored in gray separate quite nicely from the other samples. So this group is a bit more spread out. But what's also quite nice, I don't know if you can see this in the video, is that the PC1, which is this component, is quite strong, 62%, and which is exactly like separating the samples by diet. So this is quite nice in terms of the data set. So let's look at a couple of more things that you can configure for those cluster analyses here. So by default, we're using Bray-Curtis dissimilarities to calculate your PCOA, but there's also different other metrics available, and you can just select them and it will automatically refresh them and recalculate your ordination basically. And then you will see obviously the strength of those components change. For a PCOA, you always need a dissimilarity function. So you can use either of those. Bray-Curtis is quite common, so we'll use Bray-Curtis here. If you're interested in how those dissimilarities look, you can always go to the matrix and you can see the different values. So how dissimilar, for example, here is sample 1 to sample 1 is obviously 0, but sample 1 to sample 2, and this is the dissimilarity. And when you now go for a different metric, you can see this is being updated with the current values. Okay, so as I said, I think PCOAs are really important, and any microbiome analysis should have one. So there's also a 3D representation of this. Now we have those 3 coordinates, and in this case, it's probably not helping that much because PC1 was quite strong already. So yeah, by adding PC3, we're not really getting much much information extra. But again, this depends on your data set. So there are definitely going to be use cases for this. You can use, you can calculate a simple UPGMA tree. This cluster dendrogram here. So the samples seem to separate quite nicely in this representation. In the neighbor joining tree as well. I just wanted, yeah, in this neighbor joining tree as well. Okay, and then basically, yeah, so this is basically what I wanted to show for the PCOA. So I think quite nice and also really quick to calculate. Just to reiterate my previous point. So we've done the PCOA using the species counts and the Bray-Curtis dissimilarity here. And this is our separation, and 62.6% is our first component. Just to illustrate my previous point, when you select a different rank here, genus, select all leaves, and recalculate your PCOA, this will look slightly different. So we now have 62.1% different visuals. So I guess the really important thing is it will calculate those charts based on what's selected in your main window. Maybe if we go for class, so to get a bit more of a difference, select all classes here, and then we go and do the cluster analysis. Yeah, so now the first component is 72.2. So yeah, it affects your plot. So be careful that what you are charting is actually what you wanted to chart. Okay, let's cover some of the other plots or charts that are available here. What a lot of people really like are word cloud representations. So word clouds are quite nice. So the more prominent taxa, in this case classes, are basically in larger fonts. So not a very scientific representation, but in this case here, it's actually quite nice because we've sorted this by our diet groups. So the first 18 samples come first, and then the other ones, the second diet group. So, and just by looking from a distance, you can already see differences. So Bacteroidia and Clostridia are quite prominent in this first group, and then in the other group, there's other things going on as well, like those negativity codes and so on. So really nice visualization. And again, based on what we've selected here in the main window. Okay, and this principle applies to all of the charts that we have available. I don't want to show all of them. Maybe I will show the this co-occurrence plot here. Yeah, that's not very good because there's not very many classes. So let's go and choose a different, let's go to genus level, select all genera, and then do this plot again. Yeah, and now we can see the genera that occur together and the ones that are exclusive by their connection with red and green lines. And I just go quickly through the other plots here that we have. One that is quite interesting is the, do I get the right one now? The correlation plot. Yeah, so this one is also very similar. So what taxa correlate negatively or positively in this matrix? So quite a lot going on. I actually wanted to show a different one. I wanted to show the the heat map is another representation to illustrate the abundance of the taxa across the samples. And then you can adjust the color scores, color schemes, and so on. Okay, so I guess everyone can find their own favorite plots that they would like to use and to visualize their data with. What's really important again is that you're always plotting what you've selected. Just to highlight this point, if you go back to the KEGG viewer, to the KEGG tree, and you collapse at level, let's say 3, which is the biochemical pathways, you select all leaves, and then you go to the charts and do a word cloud. You're calculating the word cloud for your KEGG data now. And this looks a bit odd. So in this case, you may want to exclude this KO 1000 enzymes category maybe. Okay. So we've covered data visualization now. And if you'd like to see more or have questions, we'll address them at the end in the Q&A session. What I'd like to do now is I'd like to switch to MEGAN scripting and automating workflows using MEGAN. And this is quite a useful feature and can really help you to make your workflows more reproducible, but also more easy. So I just close the KEGG view here. And so what am I talking about? Here on the in the bottom left corner is the MEGAN message window, and we'll make this a bit larger. And in this message window, essentially, you get a printout of everything that you're doing. So you get some user information, so some feedback, but you also get the commands that were used to in your main window. So whatever I do, for example, if I select a different file taxonomic rank here, it will update this view, and then it will say what I've just done: execute, collapse, rank, order, select, rank, order. So in other words, this is a translation of my input that I've just given through the graphical user interface. And I can use this to write a script that will perform what I would otherwise do in the graphical user interface. So why is this useful? For graphical analyses, this is maybe not so useful. The case that I could see for graphical analyses is if you want to exclude a lot of taxa for one of your plots for whatever reasons, and you want to do that for each of your data sets. You may want to deselect them, and you write a MEGAN script that deselects all taxa in your different data sets and then performs your analyses. But where this really, in my opinion, is really useful is when you want to modify or export data. And I will show you what I mean. So let us quickly select all leaves and go and export this data into a tab delimited file. So, just in a CSV. There's a lot of options, and I think we've talked about this in the last tutorial in a bit more detail. So I would just write this data into a file. And okay, and just to show you, to quickly show you this file. Let's open this in a text editor. Yeah, essentially, this is just the abundance of those taxa here across samples. I should probably quickly open this in a open this in numbers because it will be nicer. Exactly. So yeah, all of your taxa, all of your samples, and then the abundance of the taxa in those samples. And why would you want to do this? Perhaps you want to do some analysis in R or any other software, and you need your data in a text file. Okay, but what's important for the purpose of demonstrating this is when you're exporting, you can also see those messages in the MEGAN message window. Now here what have we done? We selected all the leaves on the order rank. Tells us we selected 93 nodes, and then it gives us the printout of what it's actually done to export it. So it says export what CSV, and then the format, and these are essentially the settings I selected in the in this small window that we've just seen. And then it says where did you want to write this, and this is the file I saved it in. And then it said, yo, I wrote this successfully, and this is the output. Now we can use this to write a script that will basically export our data. And this is what I'd like to show you very briefly now. And in my work, I use those scripts quite regularly to export data. So for example, I would like to export the counts for 6 taxonomic ranks of the NCBI database. And I would also like to export counts for KEGG abundance data on all 4 KEGG levels. And I will show you how I do this. Now, I've prepared the script already, and we'll have a look together. So just some comments on the top, and then the first real command here is open file, and this is my MEGAN comparison file. Now, if we can scroll up here to where we open in the message window to where we opened MEGAN, this should actually be my very first command that is here in the MEGAN window, and there it in the message window, and there it is executing. No, this is the metadata, executing open file, and then this is the path to this comparison file. So this is basically the first thing I could copy paste into my script to load my data. And then I need to specify what I want to do. And this looks like quite a lot of text, but it really isn't, because it's always repeating the same things, just with different taxonomic ranks. So I collapse my backbone tree at the species level. I select them all, and then I basically have this export command that saves the data with the settings that I've used before into a text file. And then I do the same for the genus level, for the family level, for the order level, class, and phylum. And then I do the same for the KEGG pathway annotation. And I do this for the level 1, so the top level, for level 2, level 3, which is the pathway level, and then level 4, which is the enzyme level. And when I run this script, I get all of those 10 files, and I don't have to do very much to get this. And this is not just so computer scientists like to be lazy, but they're not lazy because they're lazy, but they're lazy because they like reproducibility. So when I run this script, every time I can make sure that I get the exact same output. And we're now going to execute this script. We're going to switch to a terminal window here. And okay, so I've got a small folder that contains the comparison file that we've looked at throughout this tutorial. It's got the metadata, but that's not so important for now. This is the file we've just exported. And then it has this exporting script here. And I've also created 2 folders where I want to export the counts to, so the KEGG counts and the taxonomy counts. And just to have a quick look, so the KEGG counts folder is empty, and the taxonomy counts folder is also empty. And what I'm going to do now is I'm going to run the script that I just showed you, and it's going to execute the script, load the MEGAN comparison file, and export those 10 text files, hopefully. So this is just here the path to the Java application of MEGAN. These 2 options are the options to run scripts, and then you redirect your script to, yeah, this is basically how you just redirect the input and tell it to run the script. And then I can just press enter and it runs the script, and now it opens MEGAN, it loads the data, it exports the data. So it goes quickly through all the different views that I specified, and it will do this quite quickly, so I can't even see what it's done, but reports all the output to my terminal window. So when you go through the output here, you can see the file that it's opened, and then what it's done. So select species, export species, and so on. And what we are going to do now is we're going to have a quick look if this worked. So we're going to look at the KEGG folder. Yeah, we've got the KEGG counts, and we're going to look at the taxonomy counts, and we've got them as well. So with just executing this quick script, we've generated those 10 text files. And again, if we want to do this for the next data set, we can launch the exact same commands, and it's not a problem, and it's all reproducible. I just wanted to open one of those text files quickly to show you that this worked. So we're opening KEGG level 2 with numbers, and then we have the abundance of our different pathway groups across our samples. So this has worked really well. Okay. So, and again, so if you want to generate your own MEGAN script, it can be quite tricky to start with, but it's definitely worth automating some of your workflows. So using the message window, and also using the window command syntax, when you click this, it will basically print all the commands that are valid commands for this current window. So it will tell you everything that you can do in the in this main MEGAN window, and so you can use this to start writing your scripts. All right. So I think I've covered everything that I wanted to cover in this interactive part. Let me quickly go back to the presentation.
Perfect. Thanks, Oliver.
Yeah,
This was a great overview.
Thanks, Simone. Let me just quickly go. I've got 2 summary slides, but my computer is playing up. Share screen. Yeah, you should see my presentation again, right? So I just wanted to reiterate the few take-home messages from the interactive parts. So for the data visualization bit, so there's many more charts on top of the taxonomy and CAC views, and it's worth exploring them, and you can really get a lot of information out of it. What's really important is that you use metadata, so that you import your study metadata and give your samples some meaning. And your charts always depend on the selection in your main window. So like I said, when you select all the families in the main tree, then it's going to chart your family trees, the family data. But if you select, for example, level 2 in the CAC view, it will chart that data. And for some charts, this is obvious. So when you're doing a word cloud, it's obvious that what you're plotting. But when you're doing a PCOA, it's not so obvious. So keep in mind what you're plotting. And then for the MEGAN scripts here, they're great to automate workflows and make them reproducible. This is really useful for manipulating and exploring data, exporting data. And in order to do this, you need to observe the message window to get the commands behind your inputs. You can also always highlight the current syntax that's available for your current window. And then here, this is the way to execute those scripts on a Mac and run them. I think it's slightly different on Linux.
I want to thank everyone for joining this tutorial. Thanks especially to you, Oliver, for the demonstration. And yeah, I wish everyone a great day, and you can find the replay then afterwards on our YouTube channel.